Hello friends, today I am going to discuss about staining techniques of plant and animal cells. Staining is an auxiliary technique used in microscopy to enhance contrast in the microscopic image. Stains or dyes are frequently used in biology and medicine to highlight structures in biological tissues for viewing, often with the aid of different microscopes. Stains may be used to define and examine tissues, for example, muscle fibers, liver, brain or connective tissue. Cell populations, classifying different blood cells for instance, or organelles within individual cells. Here we will discuss some common laboratory staining techniques for staining cells and tissues of plant and animals. Let us first talk about staining plant cells. Experimental preparations of temporary mounts of an onion pill. Apparatus and materials required are an onion, glass slide, watch glass, cover slip, forceps, needles, brush, blade, filter paper, safranin, glycerin, dropper, water and a compound microscope. In procedure, take an onion and remove its outermost pill. Now cut a small part from an inner scale leaf with the help of a blade. Separate a thin transparent pill from the convex surface of the scale leaf with the help of forceps and keep this pill in a watch glass containing water. Add 2 drops of safranin stain in the watch glass to stain the pill. Take a clean slide and put a drop of glycerin in the center of the slide. With the help of a brush and needle, transfer the pill on the slide. Glycerin prevents the pill from drying up. Carefully cover it with a cover slip and avoid any air bubble from entering the cover slip. Remove any excessive glycerin with a filter paper. Observe the prepared mount of the pill under the low and high magnification of a compound microscope. In observations, a large number of rectangular cells are visible. These cells lie close to each other with intercellular spaces between them. These cells are surrounded by distinct cell walls. These cells have a dark stained nucleus and a large vacuole in the center. Staining onion root tip cells. Onion roots and sprouting seeds are used to study cell division. Onion root tips are commonly used to study various stages of mitosis because the roots are easy to grow in large numbers. The cells at the tip of the roots are actively dividing and thus many cells will be in stages of mitosis. The tips can be prepared in a way that allows them to be flattened on microscope slides called squashed so that the chromosomes of individual cells can be observed. The chromosomes can be stained to make them more easily observable. Regions of onion root tips. There are 3 cellular regions near the tip of an onion root. The root cap contains cells that cover and protect the underlying growth region as the root pushes through the soil. The region of cell division or meristeme is where cells are actively dividing but not increasing significantly in size, and in the region of cell elongation, cells are increasing in size but not dividing. Preparation of onion root tip. Select a large onion bulb with undamaged disc like stem. Under a running tap, gently rub the stem to remove old withered roots and adhering soil particles. Place the bulb on a glass jar of a suitable size filled with tap water so that the base is completely immersed in water. Change the water in the jar twice daily. When the adventitious roots have grown to 2 to 3 cm long, cut and fix the distal 1 to 2 cm of the roots in a vial containing freshly prepared fixative such as acetic alcohol. Shake the contents of the vial. Replace the used fixative with equal volume of freshly prepared fixative. Allow it to stand at a room temperature for 24 to 48 hours. After 24 to 48 hours, pour off the fixative and add 70% alcohol to the vial and shake. Replace with 70% alcohol. Repeat twice. The roots are now ready for making squash preparations. Procedures for making squash. Near one edge of a clean slide, place a root tip and excise the terminal 2 to 3 mm portion. Discard the proximal part of the root. Place a drop of acetoarsine stain in the middle of the slide and transfer the terminal portion of the root to it with forceps. Use a spear needle and a scalpel to tease the root tip into a number of small pieces. With an aid of a masher, crush the root tip tissue into a through pulp so that no large pieces are observable on the slide. If necessary, add another drop of stain, but avoid excess. Drain off all the stain which sticks to the masher on the slide to minimize the loss of cellular material from the slide. Place a cover slip over the suspension of cells. Gently tap the cover slip with the rubber end of a pencil or the wooden end of a dissecting needle to spread the material. Care must be taken not to move the cover slip. Place the slide over a steam bath or warm it over a hot plate until the stain at the cover slip edges begins to show sign of drying off. Remove the slide from the steam bath and further flatten the cells by applying firm pressure from above while the slide is being held between the folds of a mat made from sheets of tissue paper. This will also expel the excess stain from under the cover slip, as well as spreading the chromosome content of dividing cells in an even layer. This slide may be used for observation for about 30 minutes without sealing. Its usefulness can be extended to 2 to 3 days if the slide is sealed off by nail varnish. Now, method of staining algae. Algal cell can be visualized better with Sudan black bee staining. In method, prepare smear of algae on a slide, let it dry thoroughly in the air and heat fix it. Flood the entire slide with Sudan black solution and allow the slide to remain undisturbed at a room temperature for 5 to 15 minutes. Drain off excess stain and counter stain with safranin for 30 seconds. Wash with water and air dry. Or algae are emulsified directly in the staining solution and the emulsion are allowed to stand at a room temperature for 15 to 20 minutes. A loopful of the emulsion is then removed from the top of the fluid and smeared with a circular motion upon a clean slide so that it dries quickly, leaving precipitated particles at the periphery of the drop. The slide is not heated. A 1% aqueous solution of safranin is now applied briefly and the smear is then washed with water and air dried. Slide is ready for observation under the microscope. Staining techniques of animal cells. Staining blood cells. Making a blood smear. A single smear can be made per slide. Place a drop of blood approximately 4 mm in diameter on the slide near the end if one smear is to be made, or at the proper location if 2 smears are to share a slide. Spread the drop by using another slide called here the spreader, placing the spreader at a 45-degree angle and backing into the drop of blood. The spreader catches the drop and it spreads by capillary action along its edges. To make a short smear, hold the spreader at a steeper angle, and to make a longer smear, hold it closer to the drop. Now push the spreader across the slide. This pulls the blood across to make the smear. This will yield a nice even smear. Staining of blood smear using Giemsa stain. Smear should be air dried and then dipped into 100% methanol. A coupling jar with a screw top is best for this. Place the slide into the slots in the coupling jar. Be sure the alcohol does not reach the frosted end of the slide. After 1 minute, the slides are removed and placed on end to drain the alcohol. They can then be placed into a plastic slide box for complete drying. A high quality Giemsa should be used. Not all Giemsa stains are equally in quality. Place a layer of stain in the bottom of glass coupling jar about 3 ml. Then add a buffer to level that will just cover the slides except for frosted ends when they are in the jar. Place the slide in the jar and stain at room temperature for about 50 minutes. Remove slides, rinse by dipping a few times into a plain buffer, then stand on end to dry. There is no need to cover slip the slides. Immersion oil can be placed directly on the smear for observing various blood cells under the microscope. Staining of blood smear using Leishman stain. Materials required: Leishman stain, needle, rectified alcohol, slides, etc. Procedure: prick the tip of the finger with the needle and take 1 drop of blood on slide. With another slide, make an angle of 45 degree, make a semi layer and let it dry. Put a drop of Leishman stain and allow it to stain for 2 to 3 minutes. Add a few drops of water and keep for 5 minutes. Wash off the stains using tap water. Allow the slide to dry and examine under the microscope. Observations: erythrocytes stain pink, nuclei of leukocytes stain deep purple, cytoplasm of agranular leukocytes stain pale blue, basophil granules stain dark blue, neutrophil granules stain purple, eosinophil granules stain red to orange, and platelets stain blue to purple. Staining sperm cell. Sperm from different species vary in size and shape. Bull sperm, for example, have paddle shaped heads. Rodent sperm have hook shaped head, and the heads of chicken sperm are spindle shaped and almost difficult to distinguish from the midpiece. Preparing slides for a basic morphology examination using nigrosin eosin stain. Many different staining techniques have been devised for examining sperm morphology. A nigrosin eosin stain is commonly used because it is effective, simple, and in addition to allowing sperm to be readily visualized, it is a so-called live dead stain, allowing one to assess membrane integrity at the same time as morphology. The technique for preparing an eosin nigrosin stain slide is as follows. Microscope slides and nigrosin eosin stain should be pre-warmed to body temperature. Pipette a drop of stain onto the end of a slide and then pipette a small droplet of semen next to the stain. Place the edge of another slide into the drops of stain and semen. Rock that slide back and forth a few times to mix the sperm and stain. Then smear the second slide across the surface of the first. Dry the slide rapidly by placing it on a warming plate or waving it back and forth in the air. Examine using a bright field microscope, typically using a 40 to 100x objective lens. Observation: the nigrosin eosin stain produces a dark background on which the sperm stands out as lightly colored objects. Normal live sperms exclude the eosin stain and appear white in color, whereas dead sperm, that is those with loss of membrane integrity, take up eosin and appear pinkish in color. Preparing slides for a basic morphology examination using hematoxylin eosin stain. Materials required: glass slides, cover slips, fixative, that is 1:1 ether alcohol, hematoxylin stain, acid water, ammonia water, distilled water, alcohol series 70%, 90% and absolute, 70% alcoholic eosin, xylene, DPX and microscope. Method: prepare a thin wet smear of sperm suspension on a pre-cleaned glass slide. The smear should be in such a way that all the sperm lie in a single focal plane. Air dry and fix in ether alcohol mixture for 20 minutes in a coupling jar. After fixation, stain the slide in hematoxylin for 4 to 5 minutes. Then the slide is transferred in acid water for 1 dip. The slide is rinsed in a distilled water and transferred to ammonia water till it appears deep blue in color. Pass the slide through 70% alcohol and stain it with 70% alcoholic eosin for 1 to 2 minutes. After staining, for permanent preparation, slides are dehydrated through 90% and absolute alcohol. Clear the slides in xylene for 30 minutes and mount in DPX. Examine under microscope using a 100x oil immersion objective. In observation: under oil immersion objective, the acrosome of a sperm is seen light blue in color, whereas midpiece and tail are stained pink with eosin. Staining cheek cell. Method: gently scrape the inner side of the cheek using a sterile spatula, which will collect some cheek cells. Place the cells on a glass slide that has water on it. Mix the water and the cheek cell using a needle and spread them. Take a few drops of methylene blue solution using a dropper and add this to the mixture on the slide. After 2 to 3 minutes, remove any excess water and stain from the slide using a blotting paper. Take a few drops of glycerin using a dropper and add this to the test mixture. Take a clean cover slip and lower it carefully on the mixture with the aid of a needle. Using a brush and needle, press the cover slip gently to spread the epithelial cells. Remove any extra liquid around the cover slip using a blotting paper. Place this glass slide on the stage of the compound microscope and view it. In observations, a large number of flat and irregular shaped cells are observed. The cells do not have a cell wall. However, each cell has a thin cell membrane. A deeply stained nucleus is observed at the center of each cell. No prominent vacuoles are observed in the cells. Staining of invertebrate cells. Sponges, that is poriferans, are very simple animals that live permanently attached to a location in the water. They are sessile as adults. There are 5,000 to 10,000 known species of sponges. Most sponges live in salt water. Only about 150 species live in a fresh water. Petrosia fisciformis is a sponge species common on the rocky shore of the Mediterranean Sea. In this phylum, the majority of the cells are organized to form the external, that is pinacoderm, and internal, that is choanodem, covering layers. Taking into account that sponges can represent a potentially valuable experimental model to study important biological processes like cell-cell interactions and cell differentiation, their cell types identification in terms of specific morphological and cytochemical features is a prerequisite to to gain deeper insight on cell functions and tissue histology. Materials and methods: sponges of marine sponge Petrosia fisiformis kept in a aerated aquaria and fed with liquefry marine at 16 degrees centigrade and 16 hours of light a day until use. Cell dissociation: dissociate the sponge cells by mechanical dissociation. Briefly rinse sponge in filtered seawater and cut into little pieces and press through a nylon mesh pore size of 100 mm to obtain a suspension of dissociated cells. Then centrifuge at 550 gram at room temperature for 10 minutes. After 2 rinses in filtered seawater, transfer the pellet for resuspension in the same medium for final concentration of 10⁷ cells per ml. Place the freshly dissociated cells under a light microscope for morphological analysis. Analysis of cells stained with different cytochemical dyes occur under the same light microscope. Cytochemical assay: fix the dissociated cells for 30 minutes at 4 degrees centigrade in glutaraldehyde 2.5% plus sucrose 1% in fresh saline water. Then washed in a phosphate buffered saline and stained according to the cytochemical methods reported below. Hematoxylin eosin staining: wash the fixed cells quickly in distilled water and maintain in Karazi's hematoxylin solution for 8 minutes. Again wash the cells in a distilled water and stain with eosin for 2 minutes, followed by a wash on tap water. Giemsa's dye: in this staining, cells are stained for 10 minutes in a 10% Giemsa solution in filtered seawater, then washed in distilled water. PAS reaction, that is periodic acid ship: fixed cells are treated with 1% periodic acid for 10 minutes, then rinsed in a tap water and stained with SIPS reagent for 30 minutes at 37 degrees centigrade to reveal sites rich in polysaccharides with red color. Toluidine blue: here fixed cells are stained with 0.1% toluidine blue in ethanol for 7 minutes at 37 degrees centigrade to evidence acid and sulfated mucopolysaccharides. LCN blue: in this process, fixed cells are treated with 1% LCN blue in 0.1 normal HCl for 15 minutes to reveal sulfated mucopolysaccharides azure color. Sudan black: this stain is used to highlight the lipids. First, dip the fixed cells into 70% ethanol for 30 seconds and then stained with a saturated solution of Sudan black in 70% ethanol for 15 minutes at 70 degrees centigrade. Again, rinse the cells into 70% ethanol and washed in distilled water, black color. Observation: the major sponge cell types are observed after mechanical dissociations of Petrosia fisiformis specimens are identified and characterized in a living and fixed cell suspensions. After dissociation, isolated cells become roundish, their diameter ranging from 2 mm to 20 mm. According to granules amount and distribution and to the presence of flagella, isolated cells can be divided into 5 different types. The following cell types also marked after observations: cells with flagella, cells without granules, cells with very small granules, cells with a few granules and with many granules. Microscopic study of animal cells, tissues and small animals may be done by temporarily or permanently mounting the specimen on the slide by several methods. Histology is the study of the structure and function of cells, tissues and organs of the body at the microscopic level. Hence, it is often referred to as cell, tissue and organ biology. In summarize the topic, it can be said that cell staining is a technique that can be used to better visualize cells and cell components under a microscope. By using different stains, one can preferentially stain certain cell components such as a nucleus or a cell wall or the entire cell. Most stains can be used on a fixed or a non-living cells, while only some can be used on a living cells. Some stains can be used on either living or non-living cells. The most basic reason that cells are stained is to enhance visualization of the cell or certain cellular components under a microscope. Cells may also be stained to highlight metabolic processes or to differentiate between live and dead cells in a sample. Cell staining techniques and preparation depends on the type of stain and analysis used. One or more of the following procedures may be required to prepare a sample. Fixation serves to fix or preserve cell or tissue morphology through the preparation process. This process may involve several steps, but most fixation procedures involve adding a chemical fixative that creates chemical bonds between proteins to increase their rigidity. Common fixative includes formaldehyde, ethanol, methanol and picric acid. Mounting involves attaching samples to a glass microscope slide for observation and analysis. Cells may either be grown directly to the slide or loose cells can be applied to a slide using a sterile technique. Thin sections of materials such as tissue may also be applied to a microscope slide for observation. Staining, application of stain to a sample to color cells, tissues, components or metabolic processes. This process may involve immersing the sample before or after fixation or mounting in a dye solution and then rinsing and observing the sample under a microscope. Thank you.